Hello and welcome to everyone joining this webinar, Generation of Recombinant Tool Antibodies to Support Cell and Gene Therapy Development. My name is Paul Royal and I manage the technical sales team for Bio-Rad's Antibody Content Division, and I'll be your host for today's event. So now I would like to welcome our speaker, Sophie Mail. Sophie is a technical sales specialist for our custom antibody service, and Sophie covers the UK, Ireland and the Nordic countries. She's been with the company for over 4 years. Prior to this, Sophie worked for an antibody therapeutics company called Iontas, and Sophie holds a degree from the University of Sheffield and a PhD and postdoctoral research experience from the University of Cambridge. Now, over to you, Sophie.
Thank you, Paul, for the introduction, and welcome to everybody attending the webinar today. I want to start with an overview. Often, our cell and gene therapy customers face the challenge that they require specialized antibody reagents to enable them to quantify and characterize proteins which result directly from the expression of a therapeutic transgene. At Bio-Rad, we hope to be able to offer a solution to this. Using our antibody phage display combined with our SpyTag technology, we're able to generate highly specific antibodies in a variety of different formats, which are able to be used immediately in relevant applications. During this webinar, I hope to expand on this solution more and explain how we might be able to provide you with useful tools for your cell and gene therapy applications. For some background, antibody phage display is the method that we use to select and generate antibodies against our customers' antigens. To do this, we first need an antibody library, and ours is called the HuCal Platinum library. It's a fully synthetic, fully human library of highly diverse Fab antibodies, which equates to approximately 45 billion different clones. Because of this, we're able to describe our method as non-animal derived antibodies. Fabs are encoded within a phage mid and expressed on the surface of recombinant phage within infected E. coli cells, providing then a link between the phenotype and the genotype of that phage. We then use phage display as a tool to select for antibodies of interest by mixing our phage library with target antigens and enriching the population of phage-expressing Fabs that will specifically bind to that target, whilst washing away non-binders. This process is described as panning. It's within this panning process that we're really able to build in a level of sophistication and guide the selection to ensure that antibodies are generated which meet the customer's specificity requirements. In this example here, we're aiming to generate Fab antibodies that recognize an antigen but not a closely related antigen, or a CRA. The strategy that we would use here is to coat a plate with the antigen of interest, shown in blue, but block our antibody phage library with a CRA, shown in grey, which is found in excess in solution. The idea here is that Fabs expressed on our phage that bind to both the antigen and the CRA will first be blocked by the CRA in solution and ultimately removed during the washing steps, while those Fabs that bind to the antigen exclusively will remain and are subsequently taken forward for further panning rounds. An example of where we had a similar project for a cell and gene therapy customer was a customer who had a gene therapy which resulted in the expression of an enzyme. The customer wanted our antibodies to be able to detect the product of their gene therapy within primate serum. As the gene therapy resulted in a human protein enzyme being expressed, it was imperative that our antibodies bound exclusively to the human form and did not cross-react with the primate version, which shared 95% sequence homology. So we performed our panning strategy exactly as described here, with the human protein acting as our antigen and the primate homologue as our closely related blocking antigen. This resulted in a successful project, generating antibody clones with high specificity to the human protein and no cross-reactivity to the primate version. It may be that the binding specificity is important to you, but you may also require antibodies that perform a function, such as the generation of inhibitory antibodies. Here we have an example of where we use the same guided selection strategy to generate antibodies for exactly that purpose. The HuCal library and phage display technology was used to isolate antibodies which were specific to the active site of the West Nile virus proteinase. The wild-type protein was used as our antigen, whereas a mutant version was used as the blocking, closely related antigen. A single amino acid change in a key residue of the active site to create this mutant allowed the phage library to be blocked, and this blocking removed members of the antibody library that bound to epitopes outside of the active site. In the top right is a graph showing the results of an indirect ELISA, and here we can see that 3 unique antibodies were screened against the wild-type, mutant, and control proteins. Strong binding, as indicated by the blue bars, is seen to the wild-type protein, but this strong binding doesn't occur with the mutant or control proteins, indicating the blocking strategy was successful. The customer then went on to use these antibodies to show that they efficiently inhibited the catalytic activity of the proteinase. This indicates that the antibodies we generated are active site specific. And the 3 graphs on the bottom of the slide show the results of an experiment where, prior to the addition of the enzymatic substrate, the antibodies are pre-incubated with the protein. And what you can see is that they inhibit the catalytic activity in a dose-dependent manner. You may have noticed in both this slide and the previous primate versus human example, we're targeting very small differences. And some of you may be thinking, would it not have been simpler to design peptides to an area of sequence difference and generate antibodies to these? We can and have successfully used peptides as antigens in our projects, but there are some caveats to using peptides in this way. Primarily, peptides are short, linear fragments and ultimately do not usually share the same native conformation seen with a protein antigen. Also, from our experience, it's shown that using peptides as antigens can result in a lower affinity antibody clone. Using our in vitro system is advantageous because it allows us to use stringent blocking, as shown by this example and the one in the previous slide. And generating highly specific, high-affinity antibodies that bind the native conformation of target proteins is something that's very difficult to achieve in an in vivo method. This slide gives an overview of our antibody generation process. Customer antigens are pooled with our HuCal Platinum library, and 3 rounds of panning will occur where we're able to introduce our guided selection methods. My role as a technical sales specialist involves having detailed discussions with customers to make sure that we understand your requirements and devise a strategy for this guided selection. This may involve a blocking strategy, as previously mentioned, or perhaps blocking with a matrix component is also required. And we can also alternate our panning antigen to ensure that a desirable cross-reactivity is achieved. The output from our panning will be subcloned into an E. coli expression vector, and a primary screen will take place. This is usually an ELISA, but we are able to incorporate other methods such as flow cytometry and bioplex assays. Clones will be sequenced, and unique clones will be taken forward for expression and purification as Fabs. The Fabs will be subjected to a final quality control. Again, this is usually an ELISA, but we are able to offer other methods, such as flow, bioplex, and even Western blotting. In as little as 8 weeks, what we're able to supply is highly specific monoclonal antibodies, anywhere between 1 and 20 per project. As mentioned, historically the sophistication was in the biopanning and guided selection, and here we offered the choice of final antibody format based on the destination vector menu that we could offer at the subcloning stage. But now we can offer versatility and flexibility in the format of the deliverables too, with SpyTag technology. As standard, we now usually express our Fabs with a small SpyTag, and this is shown as a small grey tag on the purple Fab in this diagram. This has really revolutionised the way in which we're able to offer fast and flexible downstream formatting options for our customers. In just 1 additional week, SpyTagged Fab antibodies can be converted into multiple different formats by the coupling to a required capture domain. Our capture domains enable us to produce mono, bivalent, and immunoglobulin-like antibodies with a wide range of different tag and labelling options. This has significantly sped up the process that we can format convert antibodies for our customers, as previously this would have involved subcloning, re-expression, and purification. We're of course still able to offer a full immunoglobulin conversion and expression in mammalian cells, but this process does take a little longer, up to 8 weeks. One of the great advantages of the SpyTag technology is that coupling of SpyTag to SpyCatcher is based on a protein-protein interaction. As I mentioned before, a small SpyTag is expressed on our Fabs, and this can be coupled with a range of different SpyCatchers that we have available. The reaction is fast, quantitative, and incredibly specific, creating an irreversible isopeptide bond between the tag and the catcher domain. We're able to offer SpyCatchers for monovalent formats, SpyCatchers for bivalent formats, and FC catchers for Ig-like molecules. The FCs that we have available are human, rabbit, and mouse species. In-house, we've performed extensive testing on this system, showing that there's no detriment to antibody function or sensitivity when using a SpyTag clone, when compared to the same clone in one of our previous equivalent formats. In some cases, using the SpyTag system with appropriate catcher can in fact be an improvement. As mentioned previously, we have different labels. These include PE, HRP, biotin, and even site-specific cysteines should you want to perform your own coupling reaction with a label of your choice using maleimide chemistry. One of the big advantages of this system is that labeling can be done prior to the coupling of the catcher to the Fab. This means that no labeling is incorporated into the antibody binding site, and therefore no loss of function is seen with the antibody binding. Another advantage is that a single catcher lot can be used for multiple couplings. For example, if we generated 10 antibodies in a project, if all 10 are coupled to the same pre-labeled catcher, then there's no difference in the degree of labeling between the antibodies. Therefore, when you are evaluating the material yourselves, you can be confident that the differences you're seeing are down to the binding characteristics of the antibodies, not differences in labeling. Essentially, what we're able to offer is one antibody in multiple formats in an instant. This could be really useful for customers who have multiple different requirements for their antibody clones, such as monovalent formats for affinity measurement, an Ig-like format for an ADA positive control, or any further labelled format that you might need for your downstream assay. I now want to run through a practical example of a project where we generated antibodies targeting a chimeric antigen receptor, or a CAR. The antigens were generated by us for this project, and they came in the form of single-chain fragment variables, or SCFVs, and we use these in both biotinylated and unbiotinylated formats. The unbiotinylated antigens could be used for a solid phase panning, where they're coated directly to a plate, whereas the biotinylated format can be used in semi-solution phase panning, where they are coupled to a streptavidin-coated bead. After the panning, we performed screening using ELISA, and this was done on the SCFVs in both biotinylated and unbiotinylated format, as well as screening on an unrelated SCFV. This identified 196 different hits. We were further able to narrow down this number of hits by looking at a secondary screening step using flow cytometry. Cells expressing the desired SCFV antigen showed that 75 different hits were found to bind this well. These were taken forward for a tertiary screening step in the form of a K-off ranking, done using biolayer interferometry. We sequenced the 20 best clones from these 3 screening steps, and that identified 11 unique antibodies. These went on to be expressed and purified, and final quality controls were performed using ELISA and flow cytometry. We can draw parallels between an anti-SCFV project, such as the CAR-T project I just described, and the generation of anti-idiotypic antibodies, or anti-IDs, in which we have a great deal of expertise and success. The SCFV is an antibody fragment, and our anti-ID projects generally work with antibody molecules or fragments. We're able to generate 3 different types of anti-ID, as shown in these examples with our anti-IDs in purple and our customer's drug antibody in gold. A type 1 anti-ID is paratope specific, and therefore acts as an inhibitory and neutralizing antibody. This will enable you to detect free drug within a system. We can generate type 1s by performing 3 panning rounds on the drug antibody as our antigen, and in all rounds we will use a matched isotype control antibody and serum for blocking of our library. A type 2 anti-ID recognizes epitopes outside of the paratope and therefore it's not paratope specific and not inhibitory. These anti-IDs will allow customers to detect total drugs within a system, whether it be free, partially bound, or fully bound to its target. We can generate type 2s by alternating the library panning rounds on the drug antibody alone and the drug antibody in complex with its drug target. The panning round on the complex would remove any binders to the paratope area of the drug antibody, leaving us with those that just bind to epitopes outside the paratope. As with the type 1 project, we would block the library in all rounds using matched isotype control antibody and serum. Type 3 anti-IDs are complex specific, so this will exclusively detect drug in complex with its target. To generate a type 3 anti-ID, we would perform all 3 rounds of panning on the complex of the drug antibody and its target. While using isotype match control antibodies and serum to block, we would also block our library with the individual components of the complex in an alternating way, and thus ensuring that we only generate antibodies to the complex and not its individual parts. This strategy is in fact one that we would use for all of our complex-specific antibody projects, not just anti-ID development. And this type of specificity is something that's much harder to achieve in an animal-based antibody generation system. All 3 types of anti-IDs can be used as tools for the measurement of drug molecules within clinical samples, and we routinely produce these for customers who are setting up pharmacokinetic studies for clinical monitoring requirements. In addition, type 1 anti-IDs act as essential positive controls in anti-drug antibody assays. In the case of CAR-Ts, we can make CAR-specific inhibitory antibodies using similar strategies to our anti-ID production. Going back to our CAR-T project example, I wanted to explain a little bit further one of the secondary screening steps we performed, and that's the off-rate ranking. We're able to incorporate this into our projects as it gives a really good indication as to which clones might have desirable off-rates downstream. The assumption here is that perhaps a slower off-rate at this stage would indicate a higher affinity antibody clone later on. It's important to note that this is not an affinity measurement because it's done using E. coli lysates of expressed Fabs. We are able to perform proper affinity measurements later on with purified antibodies, and this is required to generate the on rates, as we need to know the protein concentration. One other thing to mention is that these are antibodies in a monovalent format, and if you're interested in converting to a bivalent format, you may see avidity improvements because of this. The unique clones from our CAR-T project were expressed as monovalent Fabs with a small SpyTag. These were then coupled into the required downstream formats, and those were an FC catcher with a human FC creating an Ig-like molecule, and a labelled bycatcher creating a bivalent conjugated Fab with a label of interest. As standard with all our projects, we would perform a final quality control ELISA. This is done on all the antibodies produced and in all formats that they are converted to. So for this project, we could look at all the clones in Fab formats, the IgG-like format, and the bivalent Fab PE format. The ELISAs are performed against the scFv of interest, the drug target, unrelated scFvs, and a range of control proteins. And we can see that the specificity shows only binding to the scFv used as a panning antigen. What's interesting is that ELISAs in the monovalent format can be used to rank the antibodies according to their binding strength, and that we see differences from the avidity effect in the binding behaviour of antibodies when they're converted to bivalent antibodies. A secondary QC was performed for this project in the form of flow cytometry screening. Anti-scFv clones in the Ig-like format were used to screen natural killer and CAR-T cells that were transfected with the target and unrelated scFvs. This showed that our clones were able to successfully distinguish transfected cells by binding the target scFv, as well as quantifying the expression levels. I hope that I have explained now how we can generate antibodies that can provide the specificity and functions required for use in your cell and gene therapy workflows. What I now want to move on to is more of the downstream steps which could be of interest to you. This is of particular relevance to our customers that might require antibody reagents for use in release testing of their cell and gene therapy products. Because we use a recombinant platform for our production method, we can show that antibody production is highly reproducible between lots. We're able to offer enhanced QC packages such as reference lot determination, and some examples are shown here which can confirm lot-to-lot purity and specificity using methods such as titration ELISAs, SDS Phage, and size exclusion chromatography. We're also able to offer further determination of antibody stability and characterization of antibody clones. This comes in the form of thermal stability measurements. We're able to calculate the temperature at which the protein starts to unfold, and also the TM, the unfolding transition temperature, which is the point at which 50% of the protein is unfolded. Accelerated stability testing can be performed on an antibody. Within this, we look at the antibody over 3 different temperatures, 4, 20 and 37 degrees, over a period of 4 weeks. And we can compare this with a control stored at -80. We're able to look at the differences between the antibody after this treatment, and EC50 values can be calculated. Finally, we can also look at freeze-thaw testing. In this example, we use a bridging ELISA and look at an antibody after the indicated freeze-thaw cycles. So, for example, anywhere between 1 and 10. The data here showed that with this clone, the antibody could withstand a minimum of 10 freeze-thaw cycles without any loss of activity. That brings me to my summary slide. At Bio-Rad, we're able to offer a fast and well-established platform for the generation of customized antibodies to suit your needs. Our strategies are in vitro, and they meet challenging specificity requirements using our guided selection strategies, which we have a lot of experience in. Using our modular antibody platform with SpyTag incorporated allows us to provide to you rapid access to monovalent, bivalent, and IG-like formats of antibodies. And this also is enabling you to use site-directed conjugation. We can screen and test antibodies that we generate for you in a wide range of relevant applications such as flow, Western blotting, and ELISA. And I've explained to you some of the extended services that we're able to offer for further characterization of antibodies. That brings me to the end of my talk today. I want to thank you all for your attention.
Thank you, Sophie, for a very comprehensive overview. Okay, we have one question that's come in, which is, what type of antigens can you work with, and are there any types of antigens that you can't work with?
Hi, Paul. Thanks for the question. The answer to this is that we can work with a really wide range of antigens. This can range from proteins to DNA, peptides, small molecules. We can actually also work with toxins. For example, if you imagine we've done projects against ADC payloads, as well as attenuated viruses. But essentially, the requirements that we need for our antigens is that they are able to be used in biosafety level 1. And so where we are working with viruses, these would need to have evidence of inactivation. We wouldn't be able to take on any live viruses, for example. But yes, in general, most antigens that customers come to us with, we're able to accommodate those.
Excellent, thank you. Okay, the next question is, can SpyTag antibodies be used in vivo?
Okay, that's a great question. So we don't advise that they're used in vivo. The reason for this is because the Fab portions themselves are E. coli derived, so there will be endotoxin present because of this, which could cause downstream issues when used in vivo. There's also a possibility that the catcher itself might be immunogenic, as this is because it's derived from Streptococcus pyogenes. And so for this reason, they're not really suitable for in vivo applications and work better in vitro. But the idea is that we use the SpyTag antibodies for rapid prototyping. So where you have multiple clones that you need to screen, the SpyTag formats will enable our customers to find the most suitable clones, which they can then select for downstream mammalian expression and purification, which is a service that we also offer. I think it's probably also important to note that due to licensing restrictions, we can't actually use our antibodies in clinical trials in anything other than mouse. So that's something important to mention as well.
Okay, and what are the typical affinities of the antibodies you generate for your service?
So affinities really are can vary from project to project, as I'm sure you can imagine. But I'd say as a guide, we are able to generate antibodies that are single-digit nanomolar affinities to their antigen from our standard package of guided selection. If you think about it, it depends on the antigen. The more competition there is essentially for an antigen, the higher the affinity clones we will likely get out of that project. So if your target is very specific, for example, you have a single post-translational modification you're trying to generate antibodies against, I think it's less likely that there'll be as many antibodies in our library which would bind to this. So therefore, there'll be less competition essentially. Whereas if your target is a larger protein, we're likely to have much more antibodies within our library that can bind. So those with the higher affinities would compete off any of those lower affinity clones. And in this example, we might be able to reach affinities even better than single-digit nanomolar. We can always perform affinity maturation on a clone if there does need to be an improvement on an affinity of that antibody to the target antigen. And it's important to mention that when we do measure our affinities, that's done in a monovalent Fab format. So you can see improvements with an avidity effect if you're looking at a bivalent antibody. Okay,
thank you very much, Sophie. I think that's probably all the time we've got for questions. If anybody has any more, I think the best thing to do would probably be to email Bio-Rad inquiries, or you can email Sophie, or just contact your local Bio-Rad account manager and they can pass your inquiry on if you don't have our contact details. But I'm sure Sophie and the rest of the team will be happy to field any questions if they come to you later on and you think, I wished I'd asked that. So I think if you don't have anything else to say, Sophie, I'd like to thank everybody for attending, and yes, enjoy the rest of your day. Thank you very much.
Thank you, everyone.